All right, good evening, everyone, and welcome to Genomics Lite. This is the first in our infectious disease series, and today we're joined by Dr. María Duque-Correa, who will be talking to us about her work with parasitic worms. First, though, I'm going to tell you a little bit more about the campus and a little bit about how this session is going to work. So my name's Em, and I'm the Education and Learning Officer at the Wellcome Genome Campus. And with us on the call today, we've also got Fran sort of hiding in the background, helping us out with any technical issues. And we're both here to answer any sort of broader questions you've got about the campus or about these sessions. In case anyone's not used Zoom webinar before, you might spot that you can't use your microphone and you can't use your camera, but there are a couple of different ways that you can interact with us during the session if you have questions or need help with anything. So we've got a chat box on the sort of bottom right where you can say hello to us, you can ask us any general questions. There's also a Q&A function at the bottom of the screen that looks like two little speech bubbles. If you've got any specific questions for Maria as we go through our talk, if you can pop them in there, that makes it easier for us to find them later. There's quite a lot of you here today, so that'll definitely just make it quicker for us to find your questions for Maria. We've also got some polls which kind of come up on the screen like a multiple choice little quiz, and that's just another way for us to ask some questions to you. I'm aware that if people are using mobile or tablet, sometimes the Q&A and polls are a bit tricky to use, so feel free to use the chat box if the others aren't working. So I'm going to share my screen again and introduce you to the campus a little bit. So hopefully you can see this nice picture of the campus, and I have now lost my nice little chat box. So we are based here at the Wellcome Genome Campus, which is just outside Cambridge in England. Now this is a picture of the campus here. I've actually never visited campus. I started during lockdown and I currently live up here in Scotland, but usually we'd be based down here in England. Now, hopefully, if you've found the chat box, it'd be really nice if you could let us know in the chat box whereabouts in the world you're based. I've spotted from the registrations that we've got quite an international group here today. So if anyone would like to tell us where they're from, that would be really nice. So we've got one hello from London, Cornwall, Netherlands, Winchester, Manchester, Cambridge. Oh, wow, we've got lots of people. We've got a good selection from across the UK. Wonder who's the furthest away. Got lots of Cambridge and London and Dorset. So we've got quite a lot. I think Netherlands so far. Oh, if that's New York, then I think New York is winning. So
I just spotted Hong Kong, Em.
Okay, yeah, Hong Kong wins. So congratulations, Hong Kong, you have won furthest away. So yeah, it looks like most of you found the chat box. So if you've got any general questions or issues, message in there and me and Fran will try and help you out. A little bit about the campus then. On the campus, there's two main research institutes. There's the Wellcome Sanger Institute and there's EMBL-EBI. EMBL-EBI, the European Biomatics Institute, focuses mainly on data. So all the staff there work with computers and data to help us understand biology a bit better. The Wellcome Sanger Institute is a mix of what we would call wet lab research as well as computer research. And Maria's based at the Wellcome Sanger Institute, so she'll tell you a bit more about her work in a second. I'm going to try and play you a quick video. Now, sometimes my Wi-Fi doesn't like me too much for this, but it's really short and it gives you a really good overview of the campus. I'm going to stop sharing and then I'm going to reshare. And hopefully this will work well for us.
Greenwich, London, site of 0 degrees longitude. Geneva, Switzerland, home city of the CERN Large Hadron Collider. Just as the Greenwich Observatory is considered the home of time, and CERN the heart of nuclear physics, so this campus on the edge of Cambridge is forever linked with the science of genomics. The Wellcome Genome Campus is where the first animal genome of a simple nematode worm was sequenced in the late 1990s. And now, the campus is leading a revolution in genomics and biodata, bringing the prospect of previously unimagined medical breakthroughs. So, scientists from all over the world are attracted to work here. Let's see how they define what a genome is.
I would say the genome is a blueprint of all the information needed to generate a whole organism.
Every organism has its own genome.
Yeah.
just as the genome connects us with all other humans. You know, we all share much more of our code than we have that is different.
Not only between each species is the genome different, but actually each individual of that species has a slightly different genome.
It's those instructions that define how the natural world looks.
That is much better than I can explain.
So hopefully that played well enough. It's just to give you a little bit of an overview of the sort of different research that happens on campus. Before I pass you over to Maria, I'll just sort of show you a couple other things that we have. So our Your Genome website is a really good place to find resources and information about anything related to genomics. You can also sign up for our education update. I'll pop the link in the chat in just a second if you want to be updated about whenever we have new events like this. And then these are all of our sort of social media and email address if you want to get in contact with us for anything else. So I'm going to stop sharing my screen now and I'll hand over to Maria. If you've got any questions throughout the chat, pop them in the Q&A and Maria will interrupt if there's anything really specific to what you're talking about. Otherwise, we'll sort of drop in with Q&As as we go. So I'll pass over to Maria.
Hello, everyone. I'm María Duque. I'm a research fellow working at the Wellcome Sanger Institute, and I'm really excited to be here sharing with, I can see 202 participants from all over the planet and tell you today what I'm doing at Sanger using genomics to study infectious diseases. Em, can you just check that my screen is showing properly? Everything fine?
It hasn't popped up for me just yet.
Give me 1 second. Is that fine now?
Yeah, that looks like looking good.
Perfect. Okay. So my field of research are parasitic worms, and specifically a type of parasitic worm called whipworm. And this worm causes a neglected tropical disease called Trichuriasis. But before we go into the specifics of this infection and the work that I'm doing at Sanger, we wanted to start the session asking you some questions to gauge, to have an idea of what do you know about host and pathogen interactions. So we are gonna ask you some questions and Em is gonna activate a pool, and some of the other questions that are open questions, you can give us some answers through the chat. The first question will be, can you tell us what is a host? So we have these options. Is a person that invites you to his or her home? Is a plant or animal on or in which a pathogen or commensal organism lives. C is a person who introduces guests and performers like on television or radio, or you can choose D, which is all the answers before. And you can now vote.
We've got lots of votes coming in. It looks like a lot of people are voting for B and a lot of people are voting for D.
Okay, that's correct, actually.
And someone in the chat said in a biological context. So yes, I think people have spotted that they are all technically correct, but in a biological context, there might be a more specific answer.
Exactly, that's correct. And they are all correct, but in the biological context, we will refer specifically in this session to a pathogen that infects an animal. All right, we go for the second question then. What is a pathogen? And the answers are, the potential answers are, any organism or agent that can cause disease. Option B is, we can define as viruses and bacteria. C is microbes. D is bacteria, fungi, protozoa, worms, viruses, and prions. And the last option is A and D.
Looks like we've got lots of ones for A and lots of ones for A and D, which is interesting. I've got a few for the others, but mainly A and mainly D, almost a 50-50 split at the moment between A and E.
Yeah, which is true. So eventually we could say it's obviously that viruses and bacteria are pathogens, and microbes, some of them are pathogens, but not all of them are pathogenic organisms. But most importantly, when we define pathogens inside that group, we have bacteria, fungi, protozoa, worms, viruses, and prions. So it's very, very, very broad. But it has to cause a disease. That's a really important component. The next question is going to be an open question. So for this, we are waiting for you to share your answers via chat. How can we know that we are infected with a pathogen?
Someone's put fever, diagnostic tests, antibodies, symptoms generally, an immune response, inflammation, getting really sick. Our body's trying to get rid of it, so like having a high temperature or a cough. Lots of different symptoms, all through symptoms or through lab tests like a white blood cell count. Lots of different examples of symptoms, antibody tests. Yeah, lots of really good answers.
Excellent. I'm just gonna pop some of the answers I thought about, but all the ones that you have said are great. We can actually grow the pathogen from samples. So bacteria, for example, we can grow from the samples. We can also grow protozoa or fungi in the lab, and we can also grow parasites. Viruses also, although sometimes it's much more difficult. We can do stainings in the lab to identify the pathogen, and we can look under the microscope to identify the particular pathogen. We can, I guess we have all heard with the current pandemic of coronavirus about doing a PCR. So we can identify genes of the pathogens using PCR. We can evaluate antigens of the pathogen in body fluids with the symptoms you all said. And just to complete, someone else talked about antibodies. Yes, obviously you can evaluate levels of antibodies against a pathogen in the blood or body fluids from someone that is infected. So that's great, awesome. Last question of this warm-up is another open question. Which tools can you use to evaluate responses of both a pathogen and a host? So what can we do in the lab if we want to know how a pathogen and a host are interacting and sensing each other?
So got a few answers coming in. So quite a few saying looking at cells under the microscope, different sorts of blood tests, ELISA tests, stem cell manipulation, different types of sort of blood counts, so white blood counts, cytokine counts. Yeah, lots of good ideas. Maybe studying an infected tissue sample, a biopsy. Yeah, lots of excellent ideas.
Great. Wow, we have a really good public this evening. So yes, we can use in vitro models to try in the lab to recreate on a dish how the pathogen is interacting with the host. We can work with animal in vivo models, or we can have samples from patients, and which type of samples, many different types depending on when the infection is happening. So with coronaviruses, we are taking swabs from the nose and from the throat, but you can also identify, for example, coronavirus in the poo, so from different body fluids. Someone talked about using the microscope. I love microscopes, so yes, definitely. But we can use genomics, and this is something that we do lots of at Sanger, and not only genomics. We also study the transcriptome. We want to know what really the pathogen and the host are producing, and then we use proteomics too. lastly, we can use biochemical tests. Someone was talking about ELISAs, and this is totally true, to measure the production of cytokines or measure the production of proteins by the pathogen too. So excellent questions. But we say we are going to talk today about genomics and the work that we do at Sanger to study those host pathogen interactions. And specifically at Sanger, my group, the group where I work with, which is the Parasite Genomics group, is doing lots of research in many different parasitic worms. So parasitic worms, we can divide in two main groups, flatworms and roundworms. They cause horrible diseases that infect billions of people around the world. And in the last years, we have really had an explosion on genomics because our team has sequenced the genome from about 80 different species of these parasitic worms. In here, I'm just sharing with you a figure that is posting the names of those genomes from species that we have sequenced. And why is it useful to sequence the genome of all these parasites? It has several uses in basic medical and veterinary science. It enables us to do evolution studies. As we compare then the genomes of the different species, we can then group the species, as you can see here in this phylogenetic tree, by clades, for example, and allow us then to see if one species is closer to another one, if perhaps the diseases that they cause are similar or not. We use the genome also because we want to understand the gene function. We don't know much about these parasites, and it's critical that we understand the genes that compose the genome, because those are the instructions that are actually making that organism and that may be then promoting disease. We study host pathogen interactions, and this is my main area of research. But we can also use this information to study transmission dynamics, as we are doing, for example, with coronavirus at the moment. We can exactly do the same with parasitic worms and identify new variants and identify how the transmission happens. And this is very important because then we can inform governments in how they have to change, for example, treatment or other measurements to control the infectious pathogens. And we use also this information of the genomes to identify targets to develop new drugs. But most importantly, because developing new drugs takes a lot of time, is to establish if there is the generation of resistance of the drugs that we use to control these parasites, which are called helmintics. So let's drill a little bit more about what I do. I work in the parasitic worms field, but specifically on those that are infecting the intestine. And there is a group of those intestinal parasitic worms that we call soil-transmitted helminths. They cause neglected tropical diseases. This means diseases that occurs, unfortunately, in the poorest areas of the planet, in the tropical and subtropical region. These infections are affecting about 1 billion people, and mainly children, unfortunately. And they don't get so much attention as other diseases because they don't happen in the developed world, and we call them neglected because there is not much research done about those diseases. And we can name 3 specific worms that we know are intestinal parasitic worms causing great burdens. The first ones are roundworms, and these are the worms from the genus Ascaris. The second ones are hookworms, and we have 2 different species, Necator americanus and Ancylostoma duodenale. And the worms that I'm studying are called whipworms, and these are from the species Trichuria. And I'm just showing you here 2 pictures, one of a male and one of a female worm. And as I was mentioning, there is a vicious cycle of poverty and neglected tropical diseases. So where there is poverty and there is lack of sanitation and potable water, the communities are contaminated with these parasites. And then obviously there is exposure to infection. And once one gets infected, this carries disability, medical costs, stigma and exclusion, and then leads to more poverty. I wanted to introduce today a little bit more about how the infection with whipworms happens. So we get infected with whipworms when we ingest eggs that are present in contaminated food and water. And when those eggs arrive to our intestine, they hatch and they liberate little larvae that then burrows into the gut lining. In there, the larvae is gonna mold and grow and mature over 3 months until they become adults, both female and males. And these adult worms, then they mate and then they produce more eggs that are released on the feces. Those eggs, then they go through a process of generating the little larvae that we see here, which is called embryonation. And this process is dependent on moisture and warm temperature. This infection causes a lot of nasty symptoms like diarrhea, anemia, rectal prolapse, as you can see here, but it's related also to impairment on development of children. What I'm most interested in is actually to understand how the worms live inside the gut lining. So we know that in larvae, in the early infection, the larvae that comes out of the eggs, it infects the bottom of the gut lining of our intestine. And in this case, the larvae is only 100 microns long. It's completely intracellular. It makes a tunnel inside all these cells that you can see here. And then the larvae, we say it grows until becoming adults. And one section of the worm is still intracellular into the gut lining, but the posterior end of the worm is then free on the lumen of the intestine. And in this way, female and male worms can mate. And at this point, the worms have grown from 100 microns to 3 to 5 centimeters. We can actually see the adult worms with naked eye. So in here, I'm just showing you the intestine of a mouse, and you can see how this posterior end of the adult worms is being released. And here showing you a picture by electron microscopy of how the worm creates these tunnels in the intestinal epithelia. And this is a picture of a histological section where you can see the portion of the worm that is still intracellular in the gut lining, but the portion that is freely moving in the posterior end, and you can actually identify the eggs inside this female worm. We know a lot, sorry?
We've got a couple of questions just in the chat that I think links quite well to sort of the bit you're on now about how the worms get past like the early stages of the immune systems. Like how do they evade white blood cells and get to this? I can't remember if you're going to cover this in a second, so apologies if you are.
They have a battery of potential ways of counteracting the immune system. In the earlier stages, we don't know, and that's what I'm going to cover in a second. But for the later stages, the worms, they produce molecules that they secrete or excrete or secrete. We call it excretory-secretory molecules, and they can modulate the immune response actually, and then actually paralyze the immune response, and they remain in the gut, causing chronic infections for years. But we don't really know what happens on early infection, and this is why I'm studying those interactions. So we do know what happens on ingestion. We know that the eggs are hatching when they arrive to the intestine and they liberate this little larvae that burrows in here. But we have no clue of how the larvae is actually swimming to the bottom of this gut lining and invading the cells. We don't understand which ones are the host cells, specifically if they like one more cell type than the other one. And we don't know which ones are the responses. And that's what I'm studying at the moment. We have used then transcriptomics to understand what the whipworm is producing and what the epithelia is responding with. And for this, I'm just going to show you an example of one of those experiments. We have eggs in the lab that we can hatch using bleach, and this is an sterile hatching and get larvae. We have another option of hatching those eggs by exposing them to E. coli, which is a bacteria, and then we get the larvae out. But we can also infect mice and get the larvae out of the mice. And we did all these experiments and we sequenced all these larvae because we wanted to see if we can track any differences. What we found was that actually the larvae that we hatch sterilely or with bacteria are not very different to the eggs, but we really find differences when we infect a mouse. So if we compare just an egg with the little larvae after 3 hours of infection, we see that the larvae start to produce molecules that allow to degrade the mucus layer that we have on top of the gut lining and get inside the tissue. And this is showing in here by this high expression of these molecules. And when we compare between this 3-hour time point with the 24-hour time point, we have a further increase on those molecules that we call serine proteases and protease inhibitors. So this data has really allowed us to understand how a larvae that just hatches in the lumen of our gut, it can really move down through these layers of mucus and infects the cells that are in the bottom of our gut lining. Then we wanted to understand if those are the responses from the larvae, what is happening at these time points with the cells of the gut lining. And for this, we have used single cell transcriptomics. In this case, we are comparing mice that are uninfected with those that are infected. And after 24 and 72 hours of infection, we get this region of the intestine that the worm is infecting, which is called the caecum. We digest the caecum to isolate the cells from the gut lining, and then we sequence those cells to study the transcriptome of the cells.
Maria, can I just quickly ask, could you explain what a transcriptome is, just briefly?
Exactly. At the beginning of the talk, and was showing in the video people that were defining what a genome is. And we say that the genome is the complete set of instructions that come in every cell of our body as DNA. But to read those instructions, we actually have to transcribe the DNA to RNA. So when we do transcriptomics, what we are doing is not measuring the DNA, but measuring the RNA that has been transcribed at that specific time point. So we are, in a way, really looking in real time what the parasite, or in this case, the cells from the animal, are producing. We can also look at the protein level, but it's much more difficult to do at a high throughput way. So that's why we do lots of transcriptomics at the Sanger Institute. So it's different from genomics because we are not just looking at the entire DNA, it's transcriptomics because we are looking at what the organisms are transcribing out of the DNA to RNA. Is that clear, or do we need any
I think that's very clear. And Livio Anita has asked a good question as well, saying, are there any transcriptome differences when hatching the eggs in E. coli?
No, and that was very, very interesting. So we know that in the body, we do need the bacteria that we have in the gut, which is called the microbiome, so that the eggs hatch. But there is no difference caused by the hatching itself. It seems that we need the rest of the components of the intestine to really see those differences on the transcriptome. So the bacteria is opening the egg, but is not really having a specific effect on the larvae. It seems that we need the entire gut with the rest of the mucus and the epithelial cells that live in there. All right. So we then have a look at what we found in here doing this single cell sequencing. So the interesting thing with single cell is that in this case, we even go further. We are looking at every single cell that we dissociated from the gut lining. And when we look at that data, we can identify the different cell types just by looking at the transcriptome, at the molecules that are produced by each cell type. And we have many different cell types in the gut lining. We have some that we call enterocytes, which are just the cells that are absorbing all the nutrients and the water that we get when we eat. We have other cell types that are called goblet cells, which I love, and they look like a glove and produce lots and lots of mucus, which is really important to protect us against infection. We have other cell types that are called enteroendocrine that produce hormones, some very particular ones that have like some hairs that are called top cells. And these cells in here, which are called transamplifying cells, are cells that are constantly divided and will give rise to the other different cell types. And the sequencing, the single cell sequencing is so specific that we can actually identify, I'm just putting here examples of genes that we know that are expressed by the different populations, and very clearly determine if a cell is one of these different types that we find in here. But besides knowing the different cell types that the mice have, we can then now compare what happens with infection. So in here, I'm showing you a comparison between uninfected mice and infected mice between 24 and 72 hours of infection. And what we found is that this population in red in here, which looks like the nose of this face, is actually increasing with the infection at 72 hours. And this is what you are seeing in this plot in here. The size of this group has really increased with infection at 72 hours. And we are currently doing more experiments to understand what that population is producing, how it's important in starting the immune response to the worm, and how the worm is then perhaps counteracting that population so that the immune response doesn't turn on and they can live in the host for a long time.
Brilliant.
All
Okay.
right.
Cool. Maria, so we've got another great data question. So on your charts, what does UMAP1 and UMAP2 mean?
Mm-hmm. This is a way, a new map plot is looking at the different expression of all the genes that we have to determine the different populations. So we use a cluster algorithm to analyze the data of all this amount of cells. In this case, it's about 100,000 cells that we have analyzed. And the algorithm allows you then to graph the differences between the groups. So what we are looking with a new map plot is an algorithm that looks in all the sequences of all the cells and allows you to pull apart the different clusters just because of the different expression of the genes in the cells. I hope that's clear. It's very hardcore, heavy data analysis
there's
to explain.
I think you've answered it, but I'll ask again. How are these analyses made? Is it through like microarrays? But this is basically the data from the sequences that you've then used an algorithm to put together.
Exactly, it's all computational. So we do work with the mice, we do extract the cells, we then sequence the cells. We have to kind of break the cells to liberate all the RNA that we were talking about we are gonna sequence. All these sequences just come back to us when they are read as files of data. And all these analysis is just done by a computer by using an algorithm. And one of the type of algorithms that we can use to identify the different cell types is these UMAP plots. But if someone else is interested, there are other type of algorithms that generate T-SNP plots, for example. It's just showing variation in the genes expressed by the cells. All right, we are gonna move gears a little bit now. And because I have been talking a lot, I'm gonna ask you a question and hopefully you can participate. All right, if someone tells you, you will be cured of a disease by getting infected with whipworms, would you do it? You can answer no or yes.
I put a poll up again, so hopefully you can see that. And again, if anyone's having issues getting to the poll, you can pop that in the chat. But it's pretty mixed. We've got slightly more no, like about 65% no, 35% yes, but it keeps sort of wiggling around. One very good, a couple of good comments in the chat saying it would depend on the type and severity of disease. So I imagine some people might go for this if the disease that they'd get cured from is really bad, but I'm guessing that if it's just like the common cold, I wouldn't do it for the common cold.
I like the comment, especially if it was COVID. So thank you, that made me laugh.
Okay, good questions. I mean, it's a very personal question, I think, but we are going to tell you a story about a collaborator.
And I think, sorry, Maria, and someone's also said it depends on who the someone was as well.
Yes. I mean, you have to understand that when you get infected with whipworms, you need to swallow the eggs that come from the poo of someone, but we can clean them really well. And the interesting thing is that we know that in the countries where we have get rid of whipworms, like in England, the rest of the UK, Europe, and in the United States, where whipworms were, I mean, there were infections with whipworms all around the world. It's not only in the tropics. We have records of infections with whipworms from the Vikings and the Romans and from the 20th century in Denmark, for example. But we got rid of them because we have sanitation. And then we don't have those infections happening anymore. In those countries, we actually have an increase on diseases that are inflammatory, like asthma, like inflammatory bowel disease, lots of other different allergies and autoimmune diseases. So there is a hypothesis behind the eradication of intestinal worms, and is that actually we need to have worms to balance the immune responses in our body. And when we get rid of worms completely, then those diseases that are inflammatory, they flare up. And this is the reason why one collaborator of us, Dr. Peter Nesom in Denmark, who has psoriasis, which is an inflammatory disease from the skin, he actually infected himself with whipworms. But Peter is also studying whipworms himself. And the great thing of Peter being infected with these worms is that we have access to the eggs that he produces and access to adult worms. And you will say, but there are millions of people infected with these worms. So anyway, you could get those eggs and those parasites from real patients, from people that are really sick because of the disease. And the problematic is that in the endemic areas, we don't only get infected with whipworms, we are at the same time infected with other worms like hookworms and roundworms and with protozoa. So it is very difficult to have a good source of clean whipworms and to have a good source of adult worms. So we are very happy because we can do lots of experiments, and we actually use the adult worms to create a better version of the genome. And with this better version of the genome of Trichuriasis trichuria, we are actually running a public engagement project that I wanted to share with you. So we say that the genome is all the set of instructions, but on its own, just the letters, they don't tell us anything. So scientists, we have to annotate the genome. That means to identify the different genes that compose the genome and identify the different parts. So to know what is an exon or what is an intron. And to do this, you could use computers, but the computers are actually not as good as humans. And we then need to use a program where we load the sequence that we have that is predicted by the computer, but we also get data from RNA, from the transcriptome, to understand which ones are the exons, that are the parts of the genes that are actually read and transcribed, and which ones are the introns. And we create a very good models of the genes. But to do this manually, it takes ages, because if you think about the whipworm, they have around 10 to 15,000 genes. And manual curation is super difficult to do. So what we are doing is to coupling with the Institute for Research in Schools and with A-level school students here in the UK. We have already 85 schools sign up from the project. And in the last 2 years, almost half of the genes from the genome of Trichuriasis trichuria have been curated by these students. And I think that if anyone is interested in to get enrolled into this project, they can talk to Fran. All right, so this is the part of genomics I was going to talk about, but I thought it was nice also to tell you a little bit more how is working at Sanger and how my career has been changing over the years. So let's talk about working in the lab. And to start this part of the talk, I want to ask you if you can tell me what personal protective equipment, this PPE that we hear in the news, do we need to work inside the lab? Just give me answers via the chat. Exactly.
So got lots of good suggestions: goggles, gloves, masks, visors, maybe safety goggles, a lab coat, like an apron or something protective. I think people are pretty good at PPE these days. Someone put depends what you're working with, which is very good, and someone put depends on the level of lab risk, but there's lots of your classic PPE combinations of gloves, masks, and goggles.
Excellent. All the answers I had here in the presentation: lab coat, goggles, gloves, mask. But it really depends on the containment level.
There was a quality one. People are even highlighting their shoe wear, closed shoe wear, which I think is very
that's
important.
super important. Yeah, never enter to my lab with flip-flops. You will be banned of the lab forever. Yes, but it really depends on the containment level, what we call. So depending on how contagious and if we have drugs to kill the pathogen or not, we can define a laboratory as a containment level 1, 2, 3, and 4. And 1 is when you work with organisms that don't cause any disease or the disease is very mild. So if you work, for example, with E. coli, or if you work with C. elegans, you can work in a CL1 lab. If you work with Ebola, you need to work in a CL4 lab, and then you kind of need to dress as an astronaut to be able to work in a CL4 lab. If you work with tuberculosis, which I did in my PhD, you need to work in a CL3 lab, because although we have drugs to treat tuberculosis, they are not really good at it, and you can get infected by aerosols. So we were having to work under negative pressure conditions, for example, with masks all the time. It was very hard. At the moment, I'm working in a CL2 lab. In here, we use lab coat goggles, gloves, and masks. And because of coronavirus, we have other level of restrictions. But yes, it really depends on the lab. Okay, next question. Let's see what you answer. Is it okay to bring the cell phone to the lab and eat and drink in the lab? Yes and no. And I think there's a poll, yes.
So that poll should pop just for you now, but yeah, again, if the chat's easier. Interestingly, we've got everyone saying no to eating or drinking, but a little bit more mixed on the cell phone. So about 30% of people think it's okay to bring a cell phone in, but pretty much nobody thinks you should eat or drink in a lab.
All right, good. It really depends on the biosafety level that we were talking about. So I wouldn't eat and drink in the lab at all, even if you work with organisms that are fine, like the C. elegans that we were talking about. But bringing a cell phone to the lab, it really depends. In CL1 labs, the ones that have the less biosafety level, it's fine to bring the cell phone into the lab. And sometimes those labs, they combine the offices with the lab. So people are working in their computers at the same time that they are doing experiments. In my lab, which is a CL2 lab, and from CL2 upwards to CL4, you cannot bring a cell phone into the lab, because then you will have to clean the cell phone to be able to bring it out. And the other thing is that you are working with gloves, and everything you touch, and then you touch the cell phone, you will contaminate absolutely everything. So we are not allowed to bring a cell phone into the lab. But this was just to give you an idea of things that we have to think about when we go into the lab. In reality, it's not only about how we dress or what we bring in, but it's a very interactive environment. So we work as a team. We really need the other people to work together. We work around hypotheses, and we design experiments to test these hypotheses. Sometimes we can see the things with the naked eye, as adult worms that I showed you. But sometimes, or most of the times, the things that we are working with are invisible. We can work with bacteria, we can work with DNA and with RNA, and this is just liquids, and we don't really see them. We have to do repetition of experiments, because this will allow us to reproduce the results that we have, which is really, really important in science when you are testing a hypothesis. An important component of working in the lab is frustration, because there are very few eureka moments where you actually prove your hypothesis and are happy that the experiment gives you a result that you were expecting in a way. But not only that, it's difficult to get reproducibility, and you have to go through a lot of repetition and standardization when you work in the lab. But as I say, it's a very active and interactive environment, and you are all the time discussing your results with your colleagues, showing the other person the cells in the microscope, just trying to explore different ways to do the experiments together. And I'm just sharing with you some of the pictures of me working in the lab. In here, we were having a look at whipworm eggs under the microscope, and I was showing some of the worms to someone from the public engagement team, and you can see here are some of those whipworms that we look under the microscope. All right. So to finalize this talk, I wanted to share with you my career path. You may have listened that I don't have a British accent, and it's actually because I come from Colombia. So I did my biology degree in the University of Antioquia, which is in the north of Colombia. In there, I was studying 2 different pathogens. I was studying Trypanosoma cruzi, which causes a disease called Chagas disease. And this is a protozoan parasite. And then I switched to study Mycobacterium tuberculosis. And these are some of the pictures of the campus of the university and the people I was working with there. And after I finished my bachelor degree, I was very lucky, and I had an opportunity to go to Mayo Clinic in the United States. And I was actually in Arizona. There is another Mayo Clinic in Florida and another one in Rochester in Minnesota. But I got to go to Arizona and be in the desert, which is very, very, very beautiful. And in here, I actually was not working with infection. I was studying what happens with the immune system when we are old. And these are the group of people that I was working with. This was only for 1.5 years. It really gave me the opportunity to look for PhD positions. And then I got a fellowship to do my PhD at the Max Planck Institute for Infection Biology. And this was in Germany, in Berlin. I was working again with Mycobacterium tuberculosis. It was a super multicultural environment. I have friends from all over the world, from Italy, from Germany, Malaysia, Turkey, Portugal. It was really, really great. After I finished my PhD, I stayed for another 6 months as a postdoc in that lab. And then I moved to the Wellcome Sanger Institute. And I have been there from 2014. This already we told you is in the UK, in Cambridge. And again, always surrounded by wonderful colleagues and by lots of collaboration, which I love about doing science. With this, I'm gonna end my talk. Thank you very much for your attention. And I'm really, really happy to take any questions you could have.
Awesome, thanks so much for that, Maria. We've got a few questions that have popped up already, but if anyone's got any more specific questions, if you do put them in the Q&A, it's just a little bit easier for us to find them. One of my favorite questions that's come up so far is, what has been your favorite Eureka moment? You said you don't get many, but do you have a favorite when it has come up?
Yes, definitely. So I didn't talk today about some of the work I'm doing to reproduce the infection with the worms, but in a dish, not in a mouse. And for this, we use a system that we call mini guts. So we actually grow the cells of the gut lining on a dish, and then I put the worms and try to recreate the infection. And I have been working in that project from 2016, and I have tried a million different things, and the worms were never infecting the cells. I use organoids in 3D conformation. I use them completely flat in plastic. I was putting the worms in there. They were moving for a day. The next day, they were not inside the cells until I tweaked some things and changed them, and voila, the worms were in. And then we are able then to study the interactions between the worm and the cells in this dish, which is much easier than working in the mouse at the early time points.
Awesome. There's a couple more sort of about your career and how you sort of chose to go into the field, and whether you always knew, or how did you choose sort of pathogens to study?
So it's a good question. So I always love biology and genetics in a specific. Funny enough, when I was in high school in the 2000s, then we had the the human genome coming up, and I really wanted to do genomics. But it was difficult to know what to study, because in Colombia, you kind of study either medicine or biology. And many people think that when you study biology, you are just gonna become a school teacher instead of doing real science. And my mom saw an interview of a scientist in Colombia in a magazine. And she looked for her phone and called the scientist and say, I have a daughter that wants to do science but doesn't know what to do. So she actually talked to us and referred me to a molecular geneticist, and that person guided me to study biology. So I entered and did biology. And from day one, from the very first class of my biology degree, the teacher was studying Chagas disease, so this parasitic infection. And I really knew I wanted to study the immune response and parasites. So I just got rolling with that.
Awesome. I'm just sort of flicking through other questions that we've got. A nice one about sort of working in the lab. So there was one about how many people do you tend to work with in a lab? And then what's the sort of most difficult or your least favorite thing about your job?
Okay. So how many people? It really depends on the lab that you are working at. When I did my PhD, the lab we were 50 people, 50. So it was huge. It's a mixture of technicians, PhD students, postdocs. Was really, really big. When I was in the United States, we were only 7 people in the lab. So it really varies. It depends who your principal investigator, the group leader of the lab is. If it's someone that has a lot of experience, usually they have bigger labs, and they tend to be like big professors. But someone that is just starting their career, they would just start with a very small lab. For the second question about what do I like most, I love that every day you can see a completely different thing and try different things. For me, the hardest part of the lockdown is not being at home, because I can still do science from home. I have been writing papers and reading science and analyzing results. But what I miss is actually to get into the lab and looking at the cells under the microscope and doing the experiments. And I was moving all the time before the lockdown, and now I'm sitting most of the time in front of a screen. What I like least, sometimes you can spend lots of not only time, but efforts and energy doing an experiment. And then the experiment doesn't work and you don't know why, and you cannot repeat it, and you have to do it again and again and again. And sometimes you get into crazy cycles of the experiments don't work until you figure it out. So obviously it's great when you figure it out, but the time when nothing is working, it doesn't feel nice.
Awesome. I've spotted one question that I think will be a perfect sort of wrap up for the talk today. Someone's asked, what do you think the future of genomics or of sort of infectious disease research looks like? I think that's a really good sort of ending question for us.
Oh, it's very, very high moving field, I will say. I think it's with the pandemic now, it has opened the eyes for many people about infections. In a way, in developed countries, we have forgotten about infection and about the importance to infections, because we obviously have access to vaccines and antibiotics. So we have controlled infections, but there are millions of people around the world suffering from malaria, from tuberculosis, from parasitic worms. And the coronavirus just brought that notion of how bad it is to be infected with a pathogen, and with a pathogen that we don't know anything about, that we don't have vaccines, and that we can't treat. And it's here where genomics is really making a breakthrough. Specifically for us, we can give this example from Sanger. We are sequencing coronavirus at the moment. And just by sequencing the different strains that we get from people infected in the UK and elsewhere in the world, we are able to identify those variants that make us be worried, because then maybe the vaccine is going to be compromised, or maybe the drugs that we have at the moment are not going to work against this virus anymore. So it is an eye-opener. I think it's great to have all these tools fighting this pandemic under other conditions, and without the science and the genomics that we have at the moment, it would be completely impossible.
Awesome. Well, that brings us up to sort of 5:30, so we'll wrap up there. Maria, thanks so much for coming along and speaking with us tonight. Just as a reminder for anyone watching, we've got 3 more talks on infectious diseases coming up in the next few weeks. So next week, we'll be looking at what research looks like in the lab, specifically looking around malaria. And then the week after that, we've got a really interesting discussion about biosecurity. So how that sort of works. And then the week after that, we'll have a whole panel session on career pathways. So for those of you that had lots of questions about careers and sort of study routes, that might be a particularly interesting one for you. But thank you very much everyone for coming along, and hopefully we'll see you again next week.